Hello, sir, and hello, everyone. My name is Tishintana Sanjeev. We all know the human body is made of elements, tissues, organs, and systems. But do we know how much of blood is in the body? The amount of blood in a person's body depends on their size and weight. An average adult has about 1.2 to 1.5 gallons of blood. So, now what is blood? Blood has four main components, which is plasma, red blood cells, white blood cells, and platelets. Blood transports oxygen and nutrients to the cells and carries away carbon dioxide. Now, I would like to talk about red blood cells, RBC. Red blood cells, also known as erythrocytes. Approximately 2.4 million new erythrocytes are produced per second in human adults. The cells develop in the bone marrow and circulate for about 100 to 120 days in the body before their components are recycled by macrophage. RBC have the shape of a flat disc and don't have a nucleus like white blood cells. So the topic that I have chose to speak about is RBC indices. Red blood cell indices are blood tests that provide information about the hemoglobin content and size of red blood cells. It can help to determine if you have a certain type of anemia. Tools used to count red blood cells. To count the red blood cells, RBC indices, laboratories usually use automated hematology analyzers called CBC machines. The automated hematology analyzers employ technologies like flow cytometry or impedance method. These machines help to analyze blood samples and provide quantitative and qualitative measurements of various blood components like RBC indices. Flow cytometry is where a blood sample is passed through a laser beam and the scattered light and fluorescent signals are used to analyze and count the cells. Impedance is actually where a blood sample is passed through a small opening and the electrical resistance changes as the cells pass through and are used to count and analyze them. The manual method to count RBC is using the hemocytometer. A hemocytometer is a specialized microscope slide with a grid attached onto it, which allows manual counting of cells under a microscope. Blood samples are diluted before being loaded into the hemocytometer to facilitate accurate counting. The hemocytometer is used in situations where automated analyzers are unavailable or for specific applications like counting the cells in the body fluids. The example of hemocytometer is Neubauer. The first picture is the automated hematology analyzer, which is the complete blood count machine, and the second picture is the Neubauer chamber. There are four types of red blood cell indices, which is mean corpuscular volume MCV, mean corpuscular hemoglobin MCH, mean corpuscular hemoglobin concentration MCHC, and red cell distribution with RDW. First, I would like to start off with mean corpuscular volume MCV. The MCV actually measures the average size of your red blood cells. The formula for MCV is hematocrit times 10 divided by RBC count. Next is the mean corpuscular hemoglobin, which is known as MCH. The MCH measures the average amount of hemoglobin in a single red blood cell. The formula for MCH is hemoglobin times 10 divided by RBC count. Next is mean corpuscular hemoglobin concentration, which is known as MCHC. The MCHC measures how concentrated the hemoglobin is in your red blood cells. It also calculates the size and volume of red blood cells. The formula for MCHC is hemoglobin times 100 divided by hematocrit. Last is the red cell distribution width, known as RDW. The RDW measures differences in the volume and size of your red blood cells. The formula for RDW is standard deviation of MCV divided by mean MCV times 100%. These test results are in the normal range like MCV 80 to 100 femtoliter, MCH 27 to 31 picograms per cell, MCHC 32 to 36 grams per deciliter, and RDW 12% to 15%. RBC indices significance. The RBC indices significance can help to determine if you have a certain type of anemia. Anemias can be classified using erythrocyte indices in the following way. The MCV, MCH, and MCHC indicates normal normocytic, which is normal cell size, normochromic, which is normal cell color anemia. It's mostly caused by acute blood loss. The decreased MCV, MCH, and MCHC indicates microcytic, which is small cell size, hypochromic, which is pale colored cell anemia. It's often caused by iron deficiency. Increased MCV, MCH, and MCHC indicates macrocytic, which is large cell size anemia. It's often caused by vitamin B12 deficiency. In conclusion, RBC indices are essential for diagnosing anemia and other blood disorders. They help to assess cell size, hemoglobin content, and overall blood health. Understanding these values aids in proper diagnosis and treatment, ensuring better patient care and management. These are the references that I've used for my presentation. That's all from me. Thank you very much for listening.